Now in my last video I asked you for some homework and none of you did it, which is no surprise. I knew that was going to happen. But I figured it'd be nice to see if any of you would. But here's what I'm going to do now. What I'm going to do now is set out some definitions before we carry on, just so no one's confused. Okay, in science, when we talk about hypothesis, that's your guess. It's your educated guess at best, based on an observation. And once you have this hypothesis, then it goes through the scientific method of testable experimentation, peer review, their testable experimentations to come back to you, and falsification. Very important. There's more to it, but I'm not going to go deeper into it, because no doubt if you're searching through videos based on evolution or genetics or any science at all and you're a creationist, you'll have heard about this explained and again and again and again. Now, theory, which we're talking about in science, is what the hypothesis will become after it's gone through the scientific method. And a scientific theory basically summarises a hypothesis or a group of hypotheses that have been supported by a repeated testing and peer review. A theory is valid as long as there's no evidence to dispute it. Now this means that science will change its mind. Known science will change, and that's not a bad thing. That is nothing to fear, people. That's a good thing. That means we're learning. If we do not change our mind, then we're not learning. We're closing our minds and living in fear. Now, this is the one I really want to work on. Law. A scientific law. Because like I say, a theory is basically an explanation of what is going to happen. What is, of what it is. Like, theory of evolution is a theory of change. The theory of abiogenesis is the theory of beginning of life. I.e., they're saying, this is what we've discovered through multiple experiments. And this is how we say it happens. And the law is based on observations in science. A law generalizes a body of observations. At the time it was made, no exceptions have been found to a law, a scientific law. Scientific laws explain things, but they do not describe them. One way to tell a law and a theory apart is to ask if a description gives you a means to explain why. For example, Newton's law of gravity, probably one of the creationists' favourite arguments about how there can be a law and a theory. Newton could use the law to predict the behaviour of a dropped object, but he couldn't explain why it happened through a law, which is why he came up with his theory. So I just wanted to get that through to you before we started. Now then, to get started on Mendel's law of segregation. Now, like I said earlier, I've touched on Mendel with his P's in the second video, the previous one to this one. And we're going to delve in deeper now. Okay. With his law of segregation. Now, as previously said, how traits are passed from parents to offspring is through the gene transmission. And genes are located in chromosomes and consist of DNA. They're passed from parents to offspring through reproduction. And this was all discovered roughly by a monk called Gregor Mendel, previously mentioned in my video. And what we're going to go into is his law of segregation right now. Mendel, as we know, studied peas. He grew peas, and he selected 7 traits to study, one of them being colour. Now, some pea plants have green pods, and some have yellow pods. And some pea plants are capable of self-fertilization. And Mendel was able to produce true breeding plants. And by true breeding plants, I mean plants that only fertilize themselves from themselves. It's like masturbation for plant kind, but this one causes you to have a child. So a true breeding yellow pod plant, for example, would only produce yellow pods. It would never produce a green one. And likewise for the green plant, the green pod pea plant. And Mendel wondered what would happen if he pollinated the green pod plant with pollen from the yellow pod plant. And to do this, in his experiments, he referred to the two parental plants as the parental generation, or the P generation. And the first offspring that came out were called the first filial, or the F1 generation. And I'll show you this in my diagram. Now what you see here is what I've just quickly drawn, is an example of two true breeding plants. So you got the two green pods as the parent plants, and they're cross-pollinating with each other, and they create three green pods here, and again two green pods, and this would carry on throughout the generations. And the same for yellow ones here. It'd all just be yellow pods. They wouldn't mix, the colours wouldn't change. Now what Mendel did was to take the pollen of this one and the pollen of this one and crossbreed them, as I'll show you in my next diagram. Now in this diagram we've got the two true breeding plants here, the parent generation, or the P generation. What Mendel did was he crossbred the green with the yellow, and what he discovered was that the resulting offspring were all green. Okay, all the F1 generation were green. And what he did next was he allowed this generation here to self-pollinate, i.e. to breed amongst itself. So I'm going to show you that in my next diagram. Now, as you can see, we've got some F1 here, which of course is the first generation, the first filial generation from the last experiment. Okay, where you got the two breeding plants, and this is their offspring. And now, after self-pollination, we come to this one, the second filial generation, or the F2 generation, which is the third generation removed from the original parents. And you'll see that we've got one yellow and three green. Now Mendel noticed this, and he realised it was a 3:1 ratio of pod colour. And it's from these experiments that he came up with the law of segregation. Okay. Mendel's law of segregation states that allele pairs separate or segregate during gamete formation and randomly unite at fertilization. There are four main concepts involved in this idea, and they are: there are alternative forms of genes, which is number one. And this means that a gene can exist in more than one form. For example here we have a Punnett square. Remember I touched on these in the last video. Okay, of a true breeding yellow and a true breeding green. And as you can see, the green is the dominant colour by having the capital letter, and the yellow is a recessive gene. Okay, so for example, the alternative forms of genes would be that there can be either a capital G for a green pod, or a small g for a yellow pod colour. And for each characteristic or trait, this is the second one. Number two is for each characteristic or trait, organisms inherit two alternative forms of that gene, one from each parent. And these alternative forms of a gene are called alleles. Pardon me. I.e. the F1 parents, which is the offspring in the boxes. In Mendel's experiment, each received one allele from the green pod and one allele from the yellow pod parent plant. The true breeding green pod plants would have GG, which is the parent here, and for alleles for pod colour, and the yellows would have GG for small g for the yellow plant, true breeding plant there. And each resulting F1 plant, the first generation, would have GG, 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 GG. One big, one small, one big, one small, one big, one small, one big, one small. Okay. Now, number 3. Now, number 3 states that when gametes, which are sex cells, are produced, allele pairs separate or segregate, leaving them with a single allele for each trait. And this means that each sex cell contains only half the complementary genes, and when gametes join during fertilization, the resulting offspring contain 2 sets of alleles, one allele from each parent, which, for example, the sex cell of the green pod plant would have a single big G allele, and the sex cell of the yellow pod plant would have a single small g allele. And after fertilization, the resulting F1 plants would have 2 alleles of a big G and a small g. Now, number 4 is where it gets really interesting, because when 2 alleles of a pair are different, one is dominant and the other is recessive. This means that one trait is expressed or shown while the other is hidden. For example, the F1 plants of big G, small g were all green because the allele for green pod colour, G, was dominant over the allele for yellow pod colour of small g. And when the F1 plants were allowed to self-pollinate, 1/4 of the F2 generation plant pods were yellow, and this trait had been masked because it was recessive. The alleles for green pod colour are big G and big G small g. And the alleles for yellow pod colour are double small g. As I show you here in this Punnett square here, where we've got F1 and F1 alleles. So that would be from self-pollination of the F1 genome. And as you can see in the Punnett square, get the 3 green and 1 yellow. Well, the yellow's fairly blurred in that picture for some reason. Okay, and that would be the F2 generation there. Now, for Mendel's law of segregation, we see that the alleles for a trait separate when gametes are formed through a type of cell division called meiosis. These allele pairs are then randomly united at fertilization. If a pair of alleles for a trait are the same, they are what we call homozygous, and if they're different, then they're called heterozygous. The first example, which was the F1 plants, they were all heterozygous, i.e. they've got for a pod colour trait, which means that their genetic makeup or their genotype was big G, little g, dominant gene, recessive gene, and their phenotype was the green colour. Now the F2 generation of pea plants, which was the next figure I showed you, and that showed 2 different phenotypes of green or yellow, and 3 different genotypes of double big G small g, or double small g. And genotype determines what the phenotype is expressed. F2 plants that had the genotype of either double G big G or big G small g were green, and the F2 plants that had a genotype of double G small g, that is, were yellow. And the phenotypic ratio that Mendel observed was 3:1 of 3/4 green plants or 1/4 yellow plants, and the genotypic ratio, however, was 1:2 and then 1, so 1:2:1. And the genotypes for the F2 plants were 1/4 homozygous of big G, 2/4 heterozygous big G, small g, and 1/4 homozygous zygous small g. Okay, and that basically covers Mendel's law of segregation. The next one we'll be going to is Mendel's law of independent assortment. Now again, this is based on Mendel's work back in the 1860s, and it's a basic principle of genetics. Mendel formulated this principle of discovering the law of segregation. The principle states that alleles for a trait separate when gametes are formed, and these allele pairs are then randomly united at fertilization. We've already covered this. And I arrived at this conclusion by performing monohybrid crosses. which are from like your true breeding plants, i.e. one type plant, mono, hybrid, one type. These were cross-pollination experiments with pea plants that differed in one trait. For example, the example we're using is colour. Mendel began to wonder what would happen if he studied plants that differed in two traits. Would both traits be transmitted to the offspring together, or would one trait be transmitted independently of the other? From his experiments, Mendel developed a principle which is now known as the law of independent assortment. So Mendel went and performed dihybrid crosses, which is in plants that were true breeding for two traits. For example, a plant that had green pod colour and yellow seed colour was cross-pollinated with a plant that had yellow pod colour and green seed colour. And in this cross, the traits were green pod colour, big G, and yellow seed colour, big Y, are dominant. And the yellow pod colour, small g, and the green seed colour, small y, are recessive. The resulting offspring, or the F1 generation, were all heterozygous for green pod colour and yellow seed colour. And I'm going to show you this. So, we have a true breeding green pod with a yellow seed here, and that's its genotype, and that would be its gamete. Okay, the two little letters there would be the gamete. And here for the true breeding yellow pod with a green seed, we've got the genotype and the gamete here. And the F1 all green pod with yellow seed, well, that's its genotype. And I'm not going to do the gamete because there's so many variations that could be. Quite simply, at least 4 different types of gamete I could create from that. So that's what he discovered. Okay. Now, after observing the results of the dihybrid cross from the original one, Mendel allowed the F1 plants to self-pollinate, just like he did with his experimentation on colour. And he referred to these offspring as F2 generation again, but Mendel, I'll show you in a diagram in a moment, I've just got to draw it. Mendel noticed a 9 to 3 to 3 to 1 ratio, and about 9 of the F2 plants had green pods with yellow seeds. 3 had green pods with green seeds, and 3 had yellow pods with yellow seeds, and 1 had a yellow pod and green seeds. Okay. So, as you can see by my Punnett square here, we've taken the the first generation, the F1 generation, here and here, as you can tell by the genotype of dominant G, recessive G, dominant Y, recessive Y, and we've self-pollinated them. And this is the offspring that we'd get. Ignore the big black dot. So, I'm showing the colour by a stripe to make it easier, and this for the pod, and the colour for the pea is the yellow or green circle. So, a ratio, as I previously explained, was a ratio of 9 to 3 to 3 to 1. So you'd have 9 coming out like this. So 1, 2, 3.